In this video we will discuss about effect of combination modification, that is interleukin 15 cytokine additions and DAG kinase knockout on the iCAR-T-CTL. This iCAR-T-CTL is the CAR-T cells that are developed from mature T cell lineage differentiated from induced pluripotent stem cells. And we have discussed the effect on this iCAR-T-CTL individually of this interleukin 15 cytokines inductions and also this DAG kinase knockout, that is DGK alpha and DGK beta. In this video the authors presented in this video we will discuss about the results that authors has presented in the paper about the combined effect of these two modifications. So before going into the in vivo results, they have also presented their in vitro results in the supplemental figures, where they have found out that in the case during this principal component analysis, they are able to find out that the individual modifications behave differently and make a separate populations. And when they look into the genes that are overexpressed in these individual modifications, they are able to see that in the interleukin 15 inductions, they are able to see more genes that are related to DNA replications, cell cycles are upregulated, whereas the genes that are involved in inflammation response to the external stimuli are more upregulated in this DAG kinase knockout. And in the case where they use both the modifications, that is induced interleukin 15 and DAG kinase knockout, they are able to see the upregulations of the genes related to persistence and the proliferations. So it is basically the combine of both the modifications. Thereafter they did the in vivo studies in the mice, where they inject the mice with this KOC7C tumors and then inject the CAR-T cells after 3 days and used it at different conditions. And what they are able to see is that in the case where they use both the modifications, they are able to see enhanced tumor suppression, at the same time increased survival rate. And these things can be seen in the panel B, the immunofluorescence image of the tumors, and the quantifications are shown in the panel C, where we can see that in the case where they use CAR-T cells from that are derived from mature T cell lineage from induced pluripotent stem cells and are modified with this DAG kinase knockout, and at the same time they also induce the interleukin 15, they are able to see the maximum survival rate compared to all other conditions. And thereafter they also checked the persistence of the CAR-T cells or the retentions of the CAR-T cells in different conditions, and they are able to see that the similar results that they have seen for the tumor suppressions, that is more persistence of these CAR-T cells are seen in the case where CAR-T cells are derived from induced pluripotent stem cells derived mature T cell lineage, and in which this DAG kinase is knocked down and interleukin 15 is also induced. And quantifications of this are panel D is shown in the panel E, where we can see the bioluminescence of the T cells. It is high in the case when both the modifications are used in the iCAR-T cells derived from mature T cell lineage from induced pluripotent stem cells. These results are shown higher, and the results on the left hand side of the panel E is about 13 days, and on the right hand side of the panel E is about 20 days. We can see that in both the cases the consistency of the CAR-T cells remains high compared to all other cases. Now this in all together indicates that the modification, this combined modification has a synergetic effect, as we can see in the upregulations of the genes that have the benefit of both the individual modifications, and at the same time the overall effect in terms of tumor suppressions and more persistence of the CAR-T cells has also improved. So this is what they have shown in this results, that the combined modifications has an enhanced effect on has an enhanced anti-tumor response compared to the individual modifications or the base iCAR-T cells derived from mature T cell lineage from induced pluripotent stem cells. So that's all for this video. If you need to understand more about these experiments in detail and also wanted to look into the in vitro results on the supplemental figures, you can go through the reference that has been provided in the description. Thank you for your time.